we have Ms. Vijayashree. She is the senior scientist at Strand Life Sciences. She has been with Strand for over a decade. Her team has built one of the largest whole exome sequencing operations in the country, and she'll be walking us through that journey from early challenges to large scale success with real metrics and lessons learned along the way.
In 2013, focusing on mainly with whole exome sequencing and oncology tests. So during those days, handful of samples where we could process it manually, and which has been taken on to the MySic. So that is where we started with the wet lab. Since the scale up happened, like we looked at with the automation implementation in the system, so that we can cater to more samples into the extraction, and that is where it happened to make the sequencing in the evaluated sequencing platform on NextSeq. But however, with the automation which we had here, catered to only 2 batches per day, for example, because the time taken on the deck was more, and also the consumables which we are using on the deck, like pipettes, tips, and all, was a cost consuming. So that is where we came up with the medium throughput automation called Manta from Cambrian Bioworks. And with this, it could cater to more samples to feeding into the sequencing system. That is where we have a NovaSeq, where high throughput sequencing happening in the lab. So with all this workflow streamlined with the sequencing throughput, making more sequencing throughput with 263%, and also it reduced the turnaround time for processing of samples, and also reduce the cost with the operational and consumable expenses. And of course, yes, no contamination, no mix up of samples, whatever processed on the instrumentation.